Hi students, today we are going to discuss about important short notes from enzyme topic, right? Active site. Repeatedly asking question, very simple question only, but why I want to give this topic means you are having one more important 5 mark question is mode of action of enzymes. Mostly they are asking mode of action of enzymes only. For that, first you have to give about the introduction about the active site, then you have to explain about the two theories, right, for the mode of action of enzyme. Sometimes for any essay question or any 2.5 mark or 3 mark question, they will be asking active site. For the 3 mark question, this board is enough. For the 5 mark question, you have to include the two models, right? So first one is, how will you define? In an enzyme, a small portion like cleft, where the substrate binds and where the catalysis also occurs is known as active site. Okay, so an enzyme which is having two sites, that is substrate binding site and also catalytic site. So what are all the characters? It consists of two sites, that is binding site and catalytic site. Binding site means the substrate will come and attaches the enzyme part. Catalytic site means reaction site, where the reaction will take place. Next point is, in that catalytic site, the cofactors and the coenzymes for the reactions will be present. Okay. Next one is, the site is not rigid, not always rigid. It is flexible. In conformation and shape, the site is not rigid, it is always also flexible. Next one is, there are so many amino acid residues are in the catalytic site. Mostly they are coming as serine proteases. Amino acid name is serine, enzyme name is serine proteases, which is present in the catalytic site for the catalysis. Next character is, substrate bind with the enzyme through, that is the name of the bond, usually non-covalent bonds, so always it is reversible. So substrate binds with the enzyme through weak bonds only, so it is always reversible. So these are all the characters for your active site. First point, to draw the structure, one small diagram for enzyme with the active site. Okay. Suppose your question is mode of action of enzyme means, first you have to give some introduction about the active site, then you have to draw the diagram with a small explanation, 1 or 2 lines. So according to Michael-Menton theory, the enzyme combines or binds with the substrate, produces enzyme substrate complex, right, which is reversible. Immediately they will be releasing one enzyme and product. This is irreversible. Okay. So this is according to the Michael-Menton theory. Then two scientists, they have proposed two theories or models for the enzyme substrate reaction. Okay. So here, this is an enzyme with active site. This is the substrate which is ready for the binding of the active site, but the problem is, in this lock and key model which is proposed by Fisher, the enzyme active site is ready to accept the substrate which is complementary to the shape of the active site, right? Suppose the active site shape is oval, the substrate is oval, means definitely they will get attached, or otherwise the shape is different means it won't allow the reaction, because the active site structure is rigid, right? So this is the active site with the enzyme, then substrate. Suppose it allows the reaction means enzyme will bind with the substrate, right? Enzyme substrate complex. Enzyme substrate complex. Okay. Next, another scientist, Koshland, he proposed one model, that is induced fit model. This name is lock and key model. The particular key only enters into the lock. Here, induced fit model, it is always flexible. What happens is, this is the enzyme with the active site. We have some substrates for the reaction. This is the active site and catalytic site. What happens in the next reaction is, they ready to accept the substrates. They are entering into the active site and catalytic site. Here what happens is, they will go for conformational change. According to the shape of the substrate, the enzyme's active site will go for some conformational change and ready to accept the whatever may be the size or shape. Finally, the enzyme is releasing two products. So this is proposed by induced fit model, proposed by Koshland, right? So for mode of action, first you have to give active site properties diagram, then two theories. Okay.